molecules in such a way that they can maintain internal concentrations of solids that are different from concentrations in the environment. And so they need to work towards establishing a state of disequilibrium, and this requires energy input, right? And as in other types of cellular work that we've seen, primarily ATP is what supplies the energy for these processes for active transport. So one way ATP can power active transport is by transferring its terminal phosphate group directly to the transport protein, and that's primary active transport. In secondary active transport, this will be an indirect process whereby potential energy from a different concentration gradient of a different molecule is used to push another molecule. And we'll see that in just a second. So let's focus on primary active transport for the time being. So in this example, so compare with the surroundings, and a cell, at least an animal cell, has much higher concentrations of potassium ions inside the cell and much lower concentration of sodium ions than the outside. So these are pretty steep concentration gradients. And one transport system that works by this active mechanism is the sodium-potassium pump. And how does it do this? So sodium ion concentration is high on the outside of the cell, it's low inside the cell. And the potassium ion concentration is the opposite. So it's low outside the cell, it's high inside the cell. And the pump protein essentially oscillates between two conformational states. So one state has high affinity for sodium binding. So that's where we're starting here. So high affinity for sodium when the protein is in this conformation. So cytoplasmic sodium binds. And then what happens? This triggers phosphorylation by ATP. And that phosphorylation, transfer of that phosphate group, leads to a shape change. So here's the phosphate group. You can see it down here. Leads to a structural change, whereby now the protein exists in a state with a lower affinity for sodium. So sodium gets released on the outer side. And conversely, that new shape has a high affinity for binding potassium. And so potassium, which was, remember, in high levels on the outside of the cell, it binds on the extracellular side. And when that occurs, it triggers release of cleavage of that phosphate group, which then restores the protein back to its initial configuration, which, remember, had a high affinity for sodium, but actually has a lower affinity for potassium, so the potassium gets dumped off into the inside of the cell, and the cycle essentially resets. And so the pump goes through this cyclic change and conformation triggered by phosphate binding, phosphorylation reactions, and on average it moves because of the shape of the protein and the binding pockets, on average it will move 3 sodium out of the cell for every 2 potassium that get pumped into the cell. So primary active transport. And I think this is my last slide. Let's see an example of secondary active transport. So remember I said this one is a little bit more complicated. So here is the sodium glucose transporter protein. And sorry, that looks a little bit like a grainy image, but next second. So sodium is we just said is higher in the extracellular fluid, it's lower inside the cell, and that gradient here is going to provide a source of potential energy. So as sodium moves down its concentration gradient, it's going to carry molecules with it, and or push them out with it, depending on which direction it's going. So there actually exist in the body many types of sodium transporters that are coupled to active transport mechanisms, and some of them act as symporters, meaning the sodium moves another molecule in the same direction that it itself is moving. And some of them act as antiporters, meaning that it pushes in the opposite direction. So let's look in detail at this sodium glucose transporter. So both sodium and glucose actually bind here on the extracellular side, so it's the white. Sodium binds first. That causes a conformational change. It reveals a high affinity site for the glucose molecule. And when glucose binds, the conformational change again shifts and opens up a channel on the intracellular fluid side. And sodium gets released into the intracellular fluid side, which is the yellow. And so does the glucose, kind of hitches a ride and follows into the intracellular side as well. And so notice that sodium is moving down its gradient, and meanwhile the loss of sodium changes the binding site for glucose back to a low affinity site. Glucose gets released at the same time. So the net result is the entry of glucose against its concentration gradient. So just remember one point though, this sodium glucose transporter protein, this mechanism can only move glucose into the cells and follow the sodium gradient. So in contrast, on the previous slide you saw the glucose transporter protein, which was reversible. It could move the molecule in one direction or the other direction. So, for example, when blood glucose is high, the blue transporter on liver cells can bring glucose into those cells, and then during fasting, that same transporter can break down glycogen and move the glucose back out of the cells. So that's another really ubiquitous mechanism, pretty much on every cell type. All right, yes?
So that actually doesn't use ATP, right? It doesn't use ATP, does it?
It doesn't, well, it doesn't directly use ATP. It gets the energy from the other concentration gradient, but that one relies on